So already we are late. Good afternoon.
So shall I start?
Yeah.
So good afternoon to all of you. Myself, Dr. Rakesh Ahire, working as an Associate Dean in College of Agriculture, Badnapur. So all we people have gathered for the session number 1. So I will request Honorable Dr. Mangala Rai sir to chair the session. Also I will request Dr. Sanjay Kumar sir, Director, Indian Institute of Seed Science, to co-chair and convener. Also we have rapporteurs, Dr. Sangeeta Yadav. I will request Sangeeta Yadav to act as a rapporteur for this session number 1. And also we have Dr. Shivaji Maitre sir, Associate Director Seed, to act as a rapporteur for this session number 1. So I will request Maitre sir to welcome Honorable Mangala Rai sir by offering a bouquet.
इसमें टाइम मत लग इसमें
Okay sir. So I will request chair sir and co-chair sir.
So thank you,
to proceed with the function.
thank you,
Thank you
Mr.
sir.
Ahire. So without any, in fact, so already we are late. So I welcome most respected chair of this session, Dr. Mangala Rai sir. Just for the opening remark, I request him.
The opening remark is just to solicit the kind attention of all the presenters to come one by one so that he or she makes presentation. If you are crisp, then we will be able to save some time for healthy discussion at the end. That's the only opening remark to start with.
Thank you, sir. So without wasting any time, we will start with the first presentation, and that is by Madam Dadlani. Her topic is emerging seed technology for climate resilient agriculture. Madam Dadlani, please. And please stick to the time. The time for this, all this lead speaker is 20 minutes because we are already late.
Hello. Okay. A very good afternoon to all. I thank the organizers and I thank the chairperson and co-chairperson for giving me this opportunity to make a presentation on seed technology for climate resilience. हम इससे कर लें क्या? नहीं हुआ. Okay. So we are talking about the seed technologies for climate resilience, keeping in view the rising world population and the receding resources, natural resources, climate extremes which are aggravating every day, every year, affecting the agriculture directly, and also the fact that plant and soil processes are most affected by climate. And considering that the seed is always in direct contact with the soil before it germinates, and then it comes to the environment when it emerges. So almost all the processes during germination under the soil, and then photosynthesis, respiration, water relations, everything is affected both by the environment outside and below the surface, that is soil. So microbiomes, water availability, nutrient availability, and root growth, which along with GHG emissions, all the factors have to be considered. And I would like to present here some of the ways where seed technology can help in at least minimizing some of the adverse impacts. So we know that because of the biotic antibiotic stresses, we lose a significant percentage of the total yield potential, and many a times it is just about perhaps 24 to 25% of the potential which is achieved. Now, it is a challenge which is here, and we have to learn to grow more from less. As I said, the resources are diminishing and demand is increasing. We have to find ways to maximize the output from every single input. And when we come to the inputs, then seed is the most important and indispensable input. A lot has already been spoken about the importance of seed as well as the climate impacts in the inaugural address. So I will skip most of the things which have already been spoken. It is also said, I would like to reiterate again, that though the cost of seed as an input is just about 3 to 6%, let's take it about 5%, it adds to about 15 to 20% of the total yield potential. That is the single most critical input in agriculture. It is also the carrier of not only the genetic enhancement, but also the applied technologies or the supplementary technologies. And today I will focus more about these applied technologies. Because our goal is to maximize the planting value, is not just germination, the overall planting value of every seed, especially during emergence, stand establishment, early growth stages, both under optimal as well as suboptimal conditions. This is why the applied seed technology is required. So for sustainable agriculture, we have to find sustainable ways meeting society's present needs. At the same time, also see that it doesn't leave adverse impacts for the future potential in agriculture. Here, seed technology can support sustainable farming by maximizing its efficiency, water use efficiency, nutrient use efficiency, controlling pests and diseases, and so on. These technologies aim to enhance the performance of a seed lot over and above what is expected from the genetic enhancement, that is a varietal improvement, and also the prescribed quality standards of a seed regarding the genetic purity, physical purity, germination, and so on. That is one standard requirement, but over and above, how much we can achieve through applied technologies. These may complement or supplement the genetic traits and physiological performance as well, because it takes long years of research to incorporate one desirable trait genetically. And if you wish to have, for example, resistance against a particular disease, at the same time very good early germination and vigor, and also efficient nutrient management, then if you incorporate applied technologies, it makes more sense, more economical, and more efficient. It can also help simply in improving the physical attributes and providing protection against pathogens and pests. Now, that's why when we talk about seed enhancement, earlier people used to think that it is only about priming, but it is not. It is all such technologies, all such treatments between the seed harvest and up to planting, which can improve its physical purity, germination speed, vigor, plant health, and most effectively and safely apply additives and biologicals. I'll come to that a little later. So this is the objective of our seed enhancement technologies, to improve physical property, give an initial boost, which is very important in uncertain climatic conditions, provide precision in protecting the seed by effective delivery of the protectant chemicals, inoculating beneficial microbes and applying micronutrients, and providing a means to molecular signaling for modulating seed and seedling function. This is very important. This can also be achieved through endophytic microbiome. So these are basically the different kinds of seed technologies that we will be talking about. This is a simple, most talked about, most practiced technology of seed enhancement, that is priming, which advances the seed physiologically, it reduces the time to take for the seed to germinate and emerge. And, and this happens because it is now known that the transcriptome and proteome analysis have shown that primed seed actually exhibit footprints of the late maturation stages. The programming which happens during late maturation remains in the seed, suggesting that primed seeds can retain the memory of previous stress and enable protection against oxidative stress through early activation of the cellular defense mechanism. This is basically the basis of seed hardening. This is just a simple example of what happens in seed priming, what happens and how it helps in post-priming germination and growth. This is one simple example which was very kindly shared by Dr. Umarani of TNAU. This is an example of ridge code simply by priming, very low-cost priming, and I'm not going to elaborate more about that because I'm sure Dr. Umarani will present more in her presentation, because TNAU has done very good work on applied seed technologies. Many of these have also been commercialized. This is also what happens in during priming. It is the enlargement of the embryo, enlargement of the whole seed, but more importantly, the axis, the embryonic axis. Modulation, during this period, modulation of cellular processes take place. This is a molecular interaction network which is directly linked to the perception of an external stress or external environmental signal. It can be light, it can be temperature, it can be moisture. And that, along with the internal or intrinsic hormonal signals within the seed, which may be a simple nitrous oxide molecule or GA or other molecules as well. This is just example. With downstreaming germination driving expansion, these are known some results which I am presenting. And these opened up a vast potential. This kind of research opened a vast potential to modulate cellular processes through external and internal signals using priming technology. And this is the future of priming technology, which can really revolutionize the seed technologies of tomorrow. So how the new priming protocols differ from the earlier or the traditional priming? These have been, earlier it was a simple hydration followed by a dehydration. Now these are done into various stages. And this is one example I have given, the courtesy Nunhems, which use a protocol for vegetable seed priming. First a dry heat, then a bleach for surface disinfection, then a osmopriming with aeration to enhance the physiological metabolism process. Then slow drum drying and the next one is coating with biologicals. Coating can be done singularly also, but to improve the efficiency, they use biologicals, or sometimes it can be chemical fungicide as well. Finally, they do a pelleting and then coating with insecticide. So it's a multiple 5 or 6 processes which makes the seed ready to face even a lot of adverse environmental conditions at the time of sowing, emergence, and stand establishment. So these are some of the new priming agents, like we have already in the morning also, biostimulants, biologicals, nanopriming is being talked about, and slow priming. This I can skip. Biostimulants has a lot of scope for seed priming. They can both chemical and biological in nature, not necessarily only biological. Some can stimulate some of the biological processes in a very environment-friendly manner. And the best part is that these are biodegradable, so do not leave carbon footprints. However, in order to make their large-scale application, we also need a regulatory mechanism in place. And I had a discussion with Dr. P.K. Singh some time back, and I think the guidelines which were submitted for biostimulants are also being considered for seed applications. In some of the EU countries, they already have guidelines, and our guidelines are in preparation. This is a kind of biostimulants which can result in better germination and seedling growth. This is a number of seaweeds. I have given examples, and there can be several hundreds of such possibilities. The global non-chemical seed treatment use is expected to grow many folds. This is one of the areas where there has been a significant increase in the market share, mainly because now more and more countries are not really favoring GM technology or the ban in chemical pesticides. So biopesticides, biofertilizer, biological adjuvants, and PGPRs have a lot of potential as seed enhancement technology or seed enhancement chemicals. They can be grouped into plant protectants and growth enhancers, as it is already understood. And in addition to the PBS, plant biostimulants, microbial and antipathogenic treatments there's a whole range of new types of biomolecules which are emerging as cellular signaling, as I already mentioned, that this can be simply like LCOs or lipoceto oligosaccharides, or a whole range of chemicals are there, small molecules which act as signal molecules. This is one of the examples. This is Quick Roots is a trade name. It's a private sector priming biostimulant, but there are many others. Seed microbiomes, I have already mentioned. Nanopriming. Why nanopriming? Because this is less chemical load, more efficient, and also could be less cost effective. There is some improvement of seed quality by nanopriming. This is data shared by Dr. S.K. Lal of IARI Seed Technology, the work done under National Seed Project, STR, where some good results have been seen by applying nano priming of onion seeds as well as field crops, vegetables as well as field crops. These are the field trials with nanophosphorus and nano zinc treated seed under STR. And some recommendations have also emerged. Dr. Sanjay Kumar himself is there. But a lot has to be done here to standardize. It is still not yet fully standardized. In advances in nanotechnology, a lot has been done in TNAU, where they are also focusing on green nano priming technologies. I'm sure Dr. Umarani will elaborate more on this. They are using nanoparticles from onion peel extract and using nanofibers for seed coating. Now, seed coating is just a vehicle to take many chemicals or biologicals or any other additives. Simply by using a coat, the application of pesticides can be reduced 80 to 90%. If you compare it with spray, it is about 90%. If you compare with use of granules, it is about 25%. So it is definitely more cost-effective, more environment-friendly, and more human-friendly. This is again an example of polymer coating work done in TNAU, and the slide given by Dr. Umarani, that how it can help. There are some second generation coating technologies using IntelliCoats, again a private sector patented technology which can sense the changes in temperature, or there are simply the aqua holders which can sense the deficiency or moisture deficiency, and these have a lot of scope because they can absorb water and 200 to 500 times and release it when there is deficit water in the soil. Now, there are challenges in polymer coating also because microplastics is a matter of concern. And as I said, that bio nanofibers are being considered to be used in place of microplastics, and so that it is also environmental friendly. Alternatively, biodegradable substitutes are also being tested. This is another technology which does not use any chemicals, any plastic or any outside agent, by using hygienization, by steam treatment, and it is very effective. This is some work I have quoted from Benziger 22 and Forsberg 205, which has shown that it is very effective in controlling seed-borne pathogens. Another treatment which does not use any outside chemical or application of any outside agent is the use of electromagnetic treatment. A lot of work has been done on this at IARI in Seed Technology Division. It is a non-invasive physical stimulant for specific duration, which can improve the efficiency of the seed considerably, very significantly. This is some work. This is the work done by Dr. Sudipta Basu and her group at IARI, where the use of, by using magnetopriming on high and low vigor seed, a substantial and significant improvement can be seen. This is the last of such treatments. This is a physical treatment again of cold plasma. This again has a lot of potential, but again, this technology is in kind of initial stages, in the sense that it has not yet been applied commercially in a large scale, but it can help in multiple ways. The such seeds influences decontamination, germination improvement, growth improvement, modulation of gene expression, and seed coat etching for traceability. It can also decrease the contact angle and therefore improve the efficiency of the applied chemicals or any nutrient or biostimulant. So this again is a very new upcoming technology having a lot of potential with multiple ways of influencing seed performance. So ladies and gentlemen, these are some of the upcoming technologies. So what we are looking forward are third generation seed treatment. These are called third generation because now we have come a long way in the last 42 years. These are looking at metabolic advancement through priming and post-priming, prolonged shelf life, plant biostimulants for faster seedling establishment and growth, microbials for imparting tolerance against biotic and abiotic stresses and nutrient availability, and seed coatings that can act as a protective covering, carrier of active ingredients, and sensors for external stimuli. So this is a whole world of potentials available here. I would like to conclude my presentation here by paying my humble tribute to my teacher, Professor R.N. Basu, who introduced me to the fascinating world of seeds and taught me the power of innovation. He was the only agricultural scientist who was made a vice chancellor twice of Calcutta University, and an excellent teacher, excellent innovator. Thank you, ladies and gentlemen.
Thank you, madam. Yeah. So thank you for this presenting and in fact in detail about the mechanism and the applied technology for mitigating the climate change effect. So as Chairman sir has pointed out, in fact, we will take question in the last of the session. So whoever is having any doubt or any query, so please note down. We will take in the last. So next presentation is by Dr. Uma Rani, and she will be presenting on the novel seed-based solution to mitigate abiotic stress for enhanced crop productivity. Dr. Uma Rani, please.
It's not shown. So good morning to everybody. So I stand before you to present on the novel seed-based solutions that have been developed, especially in TNAU, to mitigate the abiotic stress. So this is a small thing that we would like to know before going into the subjects. The Wisconsin Ice Age, the period was 75,000 years ago, to 11,000 years ago, mile thick glaciers were over the North America. At that time, after that is 6,000 years back, this glacier started to melt, and it gifted the North America 6 quadrillion gallons of water. That is a fifth or 20% of all the drinkable water on the surface of Earth is present in these lakes, Lake Superior, Huron, Michigan, Erie, and Ontario. So thousands of years ago, the melting of the glaciers have given North America this magnificent gift. And this marks the start of climate change, maybe. So, the last glacial maximum, that was 21,000 years ago, we had a normal temperature of around only 14 degrees centigrade, which is now 26 degrees centigrade, and we expect that it will increase to 31 degrees centigrade by the end of this century. So what will be the effects of climate change? We all know we are going through it at present. So there is increase in temperature, rainfall pattern changes, then increase in ocean temperature and the sea level increases, melting of glaciers and sea ice, extreme weather events and shifts in ecosystem, length of growing season, timing of flowering, etc. So the major effects on the agricultural ecosystem will be rise in temperature, erratic rainfall, flood, drought, and then decrease in soil organic matter. So with respect to seed production, we are expecting that all the stages of the plants, like the reproductive growth physiology, flowering time, pollen biology and dispersal, stigma receptivity, pollinating agents, seed yield, seed biochemical composition and quality, everything is going to be affected because of the rise in temperature and the erratic rainfall. So what are the solutions that we can suggest? So as Madam has been discussing, seed priming, coating, and adding to that, there is one technology that has been developed recently in TNAU, seed cubes, that we will see in the coming slides. So first, with respect to seed priming, so it was started in Ellis in 1963, and it was carried forward by Henkel in 1946, and it was perfected by Heidecker in the year 1973. And it is not new to any of the seed technologists. So what difference we have made to this particular technology, that alone we will see. So unanimously, it is followed for all crops in the Western countries to ensure speed and uniformity of germination, and also to improve the actual germination percentage. And it is also very helpful to cope up the low temperature stress faced in that part of the world. So this is the technology. Maybe it is present somewhere, but we have tried mainly to bring about hands-free quick surface drying in the seeds. So this washing machine has been customized for seed priming. So this helps in automatic, when tons and tons of seeds has to be soaked, then draining becomes a problem. So in this particular modification, we take the seeds in bags and then put it in the machine, and then we fill it with water. So the priming is allowed. And when you have, the switch, it works, it helps to automatically drain the water. And when you allow it to spin, then there is surface drying, and these seeds can be easily dried under shade to bring back to the original moisture content. So 2 packs of equal quantity of seeds are taken. They are placed in the drum and filled with water for the specific duration according to the crop, which has been standardized for each and every crop. That we call it as hydro priming, but that is followed by spinning. Why we do this spinning? To remove the extra surface water in an easy manner, and you remove the bags and then you shade it. The shade dry it to bring back to the original moisture content. So this is only for the ease of priming we tried, but to our astonishment, we found that the spinning actually is far better than the conventional priming. You can see here, the control is slightly stained, but the conventional priming is fully stained, and the embryo elongation is much better in the TNAU spin priming. This is for tomato, and this is for snake gourd. Madam has already presented. So you can see the cotyledon, it is showing some veins also. And for bendy, you can see the growth of the embryo, that is the radicle has protruded much longer. And this is for radish. For like this, we have done for all the crops. So you can see this protein profile. There is one, so this area you can, that marked in the red, you can see 2 bands. So this appears only when the seeds are spinned and dried. So we expect that some special proteins appear in the late drying stages, which is responsible for bringing about the improvement in the seed quality. So this is how we have standardized for all the crops, right from tomato up to beetroot. So now the second technology we could see is the seed coating. So seed coating is much beneficial to mitigate the climate change with respect to improving the germination, seedling growth, crop growth, substituting the organic matter, and drought mitigation, as well as delaying the germination whenever it is required. So for that we have different types of seed coatings. With respect to organic matter, we can coat the seeds with the microbial coatings. Then for tackling the drought, we can use the hydrophilic polymers, and for delaying the germination, we can go for hydrophobic polymer, and for improving the germination, we can go for hormone coating, and for better seedling growth, we can go for nutrient coating, and for crop growth and protection, insecticide and fungicides are normally used. So this is the formulation which has been developed by Tamil Nadu Agricultural University. We have the brand name of TNAU Vidyapeeth. It is already being commercialized. So the composition is, it contains hydrophilic polymer, a root promoting substance as an active ingredient, and the hormone to enhance the performance of this particular formulation. So we have standardized for all the crops. You can see that it has an exorbitant effect on the plant growth, especially the root growth. This is the black gram seedlings, the coated and the control seedlings are compared over here. And this is for groundnut. You can see the growth is, especially the root is much better when compared to the control. So because of this growth of the roots, the root resistance is ensured, and maximum nutrient uptake and water uptake is also ensured. So this particular seed coating is mainly providing the drought resistance as well as improved growth of the plants, and we can get a higher yield of at least 15 to 80% across the crops. So this is how the first, it improves the speed of germination, uniformity, and then the germination percentage itself is increasing by 8 to 10%, and there is vigorous root growth, and then there is higher number of plants per meter square, which contributes a lot to improve the productivity on a whole. Other than that, there is faster plant growth, there is dense root, sturdy plants, early flowering, greater uptake of nutrients, higher tolerance to drought and heat stress, and finally the plant yield increases by 15 to 80%. These are all the actual pictures that have been presented over here. So these are all the lab studies, and it has been standardized for all crops from rice, wheat, maize, sorghum, across the crops, for all the vegetables also, tomato, brinjal, chili, everything has been standardized for this particular crop. So the third thing I would like to discuss is the probiotic seed coating. Everybody knows that the seeds are coated with our probiotics, which are live microorganisms that are intended to proliferate in the rhizosphere and support the plant growth. So normally we coat with our Pseudomonas or Bacillus and things like that. So this is very common, and pectin and gelatin are best used as the carrier for the fungal coating. Other than the probiotic, the new thing that we have been experimenting for a few years is the prebiotic coating. That is one disadvantage of having the probiotic coating is how the colony forming units are retained when it is taken to the field. And when you sow it in a field, whether the organisms which are present in the soil naturally or natively empowers the coating which has been provided, microbes which are provided through the coating. That is one question. There are many reports which are for and against that. So we wanted to try a prebiotic coating, that is the nutrients that beneficially affects the seeds by selectively stimulating the growth and activity of the native soil microbes. So we are not adding any microbes to the coating, we are just adding the nutrients that could stimulate the native soil microbes in the spermosphere to improve the soil health. So the results are like that. We can see that we have taken in 4 concentrations this particular prebiotic coating, 4 grams, 6 grams and then 8 grams. The highlighted one is the control. Compared to that, the spermosphere microorganisms are found to be better in all the dosages. And with respect to the vigor index of the black gram, the coated seeds are much better when compared to the uncoated seeds. So this is how the rhizosphere microbial count has increased because of the prebiotic coating. Just remember, this is not the microbial coating, this is only a coating with nutrients which will stimulate the microbes. So we have studied the metagenomic studies, that is the microbial diversity has been much higher in the coated seeds rhizosphere when compared to the control seeds. This is Fisher alpha diversity and the other one is the Simpson diversity. Then the studies on the top 10 enrichment genus, we could see that in the control, Bacillus species was in the higher percentage. However, when it is coated with our prebiotic nutrients, the candidate species has been in a large percentage. So the importance is the active participation of candidates in the carbon and nitrogen cycle, and it also is involved in the stress mitigation. So because of all these aspects, we could see a very good improvement in the plant growth in black gram when the seeds are coated with the prebiotics. So the next one is the other coatings which are normally found in the literature. So the polysaccharide coatings are found. The alginate is mostly used. This alginate is known for the water retaining capacity and as a carrier of microbes. Then chitosan is also used. So this chitosan is extracted from fish, insects, prawn and crab, and it has a very good chemical reactivity. So you can add any substances, other active ingredients also to the formulation which is made out of this chitosan, and this is also useful as the seed disinfectant. Then the other coating is guar gum coating. This is known for the water retaining capacity and it's also known as the soil conditioner. Then nano coatings, in our university, Dr. Raja has come out with nano fiber coating which can be used for electrospinning as well as for wrapping the seeds. So this is also used as a carrier for the microbes and for any chemicals like zinc oxide, the nano chemicals also. So this is widely talked about, the hydrogel coating with the super absorbent polymer. Everybody knows that it is acts as a water reservoir and it acts as a remediation agent for the soils. So this is the final technology I would like to discuss. So the seed cube technology, which has been developed as first of its kind maybe. So the inspiration is the seed balls. Many of the people, especially in Tamil Nadu, they are making the seed balls, that is taking the seeds and then enclosing in soil and FOM mixture. So we wanted to improve this technology with respect to the quality of the media used and then the quality of the seeds used, and it has been published in the Hindu also as a one cube revolution. So this is the procedure that we follow. So first we coat the seeds with our own formulation and then we do the priming according to the species and we prepare the media according to the standardized ratio and then we make the cubes. So this is the small prototype that we are using. The seed cube is prepared by enclosing high quality seeds in superior growth media which is rich in organic matter. So the seeds are kept inside the growth media and then it is compressed with this machine and then it is ejected out of this particular machine prototype, and it is like cuboid shape, and now we are manufacturing, or we are producing the seed cubes for the vegetables like tomato, brinjal, chili, gourds, etc. And we are selling in the name of TNAU NutriVeg seed cubes. So each box will contain 7 to 8 vegetables. In each of the cubes there will be 3 to 4 seeds. So one cube can be kept in one pot and then watered so that it can sprout and produce the plants. So the advantages are the high organic matter. We are adding the biostimulants, we are adding bioprotectants, organic manures are good for fertility, and then there is lower bulk density and higher porosity. It increases the aeration for the germinating seeds, and it also helps in surface sowing, which is amenable for both manual and then mechanical sowing. So this has been taken to the field. So a farmer, he has grown the ridge gourd and then bitter gourd seeds with the seed cubes. So we can see that in the initial stage itself, the TNAU NutriVeg seed cubes are far better in growth when compared to the control, both in ribbed gourd as well as bitter gourd. So the farmer reported that this twining, that is, they have only after, that is 5 days earlier, they could take this process of helping the, twining process, that one. So this is the difference. So first one is the seed normal method, and the rest are the cube method. There is a 1 feet scale over there. Almost 2 to 2.5 times there is increase in growth when compared to the normal seed sowing method. Both in this is observed in both the crops. So this is even against the abiotic stress like the virus also, the TNAU seed cube had better tolerance when compared to the control seeds. This is 40 days after sowing, and this is the farmer. So what are the benefits? We can go for direct surface sowing, because these days in urban people do not know at what depth you should sow, how much water should be added. Therefore, here, since the seeds are pre-sown, we can keep on the surface itself and then water it. So direct sowing on the surface is possible. Then it has some resistance towards biotic and abiotic stress. The emergence is uniform, and it's also quick, early flowering, and higher yield is obtained. So there is warm reception among the customers. We have also seen people in the newspapers also, it's appearing. So seed-based solutions, finally, seed priming, coating, and cubes can be better used to overcome the drought, the low soil organic matter, as well as the low fertility. Thank you so much.
Thank you, Dr. Uma. Thank you, Dr. Uma. Yeah. So next speaker is Dr. B.M. Khadi. Now I invite Dr. Khadi to speak on maintenance of genetic purity of varietal and hybrid seeds in often cross-pollinated crops with a special reference to cotton. Dr. Khadi, please.
Yeah. Good afternoon to everybody. Respected chair and co-chair, dignitaries and fellow delegates. I have been asked to talk on the maintenance breeding, especially in often cross-pollinated crops. As you know very well that cotton is an often cross-pollinated crop, and I thought of talking about how we maintain the varieties and the parental lines of hybrids in cotton. Actually, the quality of seeds, apart from the development of improved crop varieties, efficient system of timely supply of quality seeds are important to get have the increase in the agriculture production and productivity. And also, already it has been talked that the contribution of quality seed alone is going to be the 15 to 20% for the production. That's what has been told. Availability of the good quality seed is essential in the view of emerging biotic stresses and also climate change. Main activities of plant breeder are three. That is, the one is, we say that the varietal development. Apart from that, seed multiplication is also the, duty of the breeder, especially the nucleus breeder 1 and breeder 2. And the third one is the maintenance breeding. It used to be there, order of the day, the maintenance breeding, but nowadays, I think it is not being followed. Especially the maintenance breeding, that is maintaining the genetic purity of the variety or hybrid parental lines to avoid the deterioration and also increase the lifespan of the variety. These are the two important things, when you go for the maintenance of the varieties or the parental lines. These are the different categories of seeds. I don't want to go into these things because these are all very basic: nucleus, breeder, foundation, and certified seeds. The maintenance breeding is the foundation to the quality seed program. Without proper maintenance breeding of varieties, varieties deteriorate rapidly in terms of their genetic potential and lose much of their value for cultivation. This is happening, nowadays in a number of varieties and hybrids because we are not taking up the, maintenance breeding at all. And it is, key to the purification and stabilization and also enhance the productive life of the variety. It is not only the job of the breeder and also the seed technologist, have to take care of the things, because it is combining the art and science of the plant breeding with the applied aspect of the seed production is the needed for the maintenance of plant varieties. The advantages already I spelled out, that is, it prevents genetic deterioration and prolong varietal and hybrid lifespan, and it helps in purification of improved cultivars and parental lines of hybrids. And the third one is the quality seed production and in turn the higher crop yields. These are the three advantages. There are some limitations also because in maintenance breeding procedure we require to test a lot of experimental material. We have to evaluate. Large number of single plants have to be evaluated in terms of agronomic performance. In such cases, only the limited number of cultivars can be handled. If there are more number of varieties and hybrids, then it will be a bit difficult. Precision method require more time, that is 2 to 3 seasons for evaluation, and we mostly depend on the phenotypic performance. That is again one more limitation. Maintenance breeding procedures are used for the varietal purification. Hence, chances to evolve new variety through the maintenance breeding. Sometimes people think that through maintenance breeding we can evolve the varieties. No, it is not. Generally, from the nucleus seed, we grow the nucleus seed and to produce the breeder seed. During the breeder seed production, we go for the selection of about 500 to 1,000 plants. That is on the basis of the morphological characters. We do that, and then we grow the plant to progeny rows, and we reject the substandard progenies. Only the best progenies are taken, and they will be bulked and given as a nucleus seed. This is a procedure is followed, but it is not a correct procedure to maintain the the productivity and also the quality of the particular variety or the hybrid. And the classification, I don't want to go into the this classification, self-pollinated or cross-pollinated, because my topic is on often cross-pollinated. Often cross-pollinated crops are the self-pollinated ones, but there is a cross-pollination from 5 to 30%. Such cross species are called as often cross-pollinated. That's what it has been mentioned. There are number of crops are often cross-pollinated. That is cotton, sorghum, tobacco, red gram, safflower, and others. As far as cotton is concerned, cotton is a predominantly self-pollinated crop, though cross-pollination is not ruled out. Cotton plant has an apparatus for encouraging the cross-pollination as well as self-pollination. Because cross-pollination is, that is mainly because of the showy flowers, flower color, right from the light cream to the light yellow, deep yellow, and also the purple color attracts the insects, especially the honeybee. So that's why the cross pollination happens. Secondly, the leaf and flowers are having the nectaries. These nectaries, attract the honeybees, and because of that, there is a cross pollination also. And third one is the pollens are spiny and also sticky, so it is very easy to carry this. And it has been observed in cotton 2 to 5%. That is a minimum, and also even up to the 81% has been reported in the cotton. The cross pollination happens sometimes. Cotton also has an effective method of self-pollination. You can see the structure of the staminal column and also the stigma. There is a proximity. They are together, so there is a possibility of having only the self-pollination. And the pollens are completely self-fertile. That is another thing. And third one is the pollen grains are heavy, so they cannot be carried by the wind. That's why there is a self-pollination. So these are the two characters. Because of that, it is often cross-pollinated crop. The maintenance breeding in cotton is an often cross-pollinated crop. As I told, heterosis has been commonly observed, and inbreeding depression is noticed to the extent of cross-pollination. Because especially the Simpson and also Duncan in 1953, they reported continuous selfing is going to reduce the yield of the varieties to the extent of 15%. And also Meredith in 1979, he ascribed poor performance of the varieties after some years. That is mainly because of inbreeding depression, which is due to the continuous selfing during their maintenance. Hence, I think in often cross-pollinated crop, there is a need of a specific method of maintenance breeding. And as far as cotton is concerned, we had almost the 3 decades N&F scheme, which was sponsored by the government of India, almost in 9 states, 3 each in South, Central and North India, with 15 centers. Almost it was there, 3 decades. 100% funding was there, human resource was there. Because of that, the cotton varieties earlier maintained very well, even the hybrids. Actually, the state agriculture universities were looking after the production of the nucleus seed, and the foundation seed was by the state Department of Agriculture. That was the system, and it was there for almost 30 years. Really, it helped in maintaining the genetic purity of the varieties systematically, and also the hybrids also. So the varieties involved in this scheme, N&F scheme, ruled long, even 50 years. I can say here from our center, 2 varieties, Lakshmi and Jaidhar, they ruled, 1950 release, but they ruled even up to the 2000. The varieties, because they were maintained very well. There are number of examples, because the scheme was run in all the 3 zones, North, Central and South, where these are the different varieties, AK235, G22, Kandwa, Badnavar, Lakshmi, Jaidhar, like that. And also the hybrids H4, DCS32, NHS44, JKHY1 and DHH1, like that, number of varieties and hybrids were involved in this scheme, and they lived longer, that I can say. Regarding the maintenance of varieties is concerned, generally the breeder seeds we plant with isolation of about 50 meters. In that, I think we have to select the individual plants, maybe 500 to 1,000, having the similar the notified morphological characters, maybe plant stature, leaf color, leaf shape, flower petal, pollen color, number of monopodia, so and so forth. These tagged plants are generally, self, and also individually they are harvested, selfed as well as the unselfed seed cotton separately. And these tagged plants are analyzed for mainly in cotton. We look for the 3 or 4 characters. But it all depends on the different crops. We have to fix the characters. Here, we go for the total seed cotton yield per plant. And the second one is the bolate. That is also very, very important. And the ginning outer and the fiber length. That is a halo length we call. These 3-4 characters we analyze, and the high yielding plants we select. Maybe about 200 plants are selected, which are having the high seed cotton yield per plant, but they should also possess the notified bole weight, ginning outer, and also the fiber length. That is on the basis of the, we find out the standard deviation and standard deviation plus or minus. On the basis of that, we select the about 200 plants for the, next cycle of maintenance. In the first cycle of maintenance, we grow all these 200 plant to progenies in 2 replications, and similarly, again, maybe 1 or 2 rows, we will have, and breeder seed bulk as a check, we keep it, and they will be tried in a replicated trial, and also the roguing, and also rejection of the progenies and the plants also has to be done during the growing season. Again, we have to look for the morphological characters which are notified for the varieties, and also shelving has to be done in the plant progenies, and then we have to analyze the shelved as well as unshelved seed cotton yield, the total seed cotton yield per the progeny, bole weight, ginning outer, and halo, same characters we are going to look for. And again, on the basis of the high seed cotton yield per progeny, and statistically non-significance of bole weight, ginning outer, and halo length. This is just to maintain all the quality characters of the particular variety. We have to analyze statistically, and the progenies which are having the non-significance, only such progenies, maybe about 50 to 60, have to be selected, and they will go for the second cycle. Generally, we go for 1 or 2 cycles, or sometimes 3 cycles also. And again, these about 50 progenies what we have selected in the first cycle of maintenance breeding have to be again replicated. Trial has to be done with the 50-meter isolation, which is recommended in cotton. And same thing is followed here as followed in the cycle 1. But the difference here is we are going to look for the more characters, because cotton is important of importance is there for the fiber quality. So we look for the total cotton seed yield, total seed cotton yield per progeny, bole weight, ginning outer, seed index, lint index. These are the very, very important characters. And as far as fiber quality traits are concerned, we call 2.5% span length, micronear, fiber strength. These are the traits I think we have to measure, and then we have to select maybe 10 to 12 progenies from this, having high seed cotton yield per progeny, and statistically non-significant for bole weight, ginning outer, seed index, lint index, 2.5% span length, micronear, fiber strength to be selected in for the third cycle of the evaluation. This is very important because these are the important quality characters have to be maintained in cotton. In other crops, I think we may have to assign the different characters on the basis of the importance. So it will go for the third cycle, but here generally we go for the second cycle 4 rows along with the again a breeder seed bulk as a check. In the third cycle, hardly there are 10 progenies. We have to go for the 3 replications minimum, replicated trial, and 10 progenies selected from the second cycle of the progeny have to be planted for the third season. That is 8 to 10 rows each, with again a breeder seed bulk in a replicated trial, and again roguing and rejecting the progenies on the basis of the morphological characters and other traits have to be done. And again we are going to have the similar characters, what I said in second cycle. Here also we are going to look for the total seed cotton yield per progeny, bole weight, ginning outer, seed index, lint index, and fiber quality are concerned 2.5 span length, micronear, fiber strength. But here we are having hardly 10 progenies, so we go for the spinning performance also, because that is most important. Because if it is a 40 counts spinning variety is there, it has to spin for the 40 counts only. It should not be less or it should not be more. That's why we go for that. On the basis of this, we select 2 to 3, maybe 4 progenies having the high seed cotton yield and statistically non-significance for all these other characters. Maybe bole weight, ginning outturn, seed index, lint index, and also the fiber quality characters like fiber length, fiber strength, micronear, and also the spinning performance. I told you it should be the same. Such things will be, such progenies will be selected, and those progenies will be bulked. And that bulk will become the nucleus seed. And then it can go for the hybrid seed. So in this way, we can maintain the, the production and the productivity as well as the, fiber quality and other morphological characters. That's how we do it. This is how the things are done. This is a flow chart. What I told has been mentioned here. As far as the parental seeds of the hybrids is concerned, again, female and male, we have to do it independently, separately, maybe 2 cycles or maybe 3rd cycle. In this, the female seed parent in the 3rd cycle, again, maybe 10 or 12 progenies, we are going to have replicated in female separately and male separately. But here, the only difference is during the 3rd cycle, we are going to produce the F1 hybrids between the female and male. That is most important because we have to maintain the productivity and characters of the hybrid. For that, we have to cross and produce the hybrid. If we are having the 10 female parents and 10 male parents, we are going to have 100 hybrids. Those 100 hybrids have to be, again evaluated in replicated trial, for the characters, what I said in the varietal things. Same thing has to be done here. And here, most important is we have to find out the heterosis and specific combining ability. The hybrids which are more heterotic and having the specific combining ability have to be selected. And you have to trace back the progenies, female progeny and male progenies, which are the things of those selected hybrids. And those progenies, again, you have to analyze on the basis of the 3rd cycle characterization. And the one which are having the non-significant results, 2 to 3 progenies, you can bulk as a female parent, and 2 to 3 progenies, maybe 4 progenies in case of male, you have to bulk, and those will become the nucleus seed of female parent and nucleus seed of male parent. So likewise, hybrid parental lines have to be maintained. Otherwise, it is very difficult. And nowadays, we are seeing that most of the hybrids, they are losing their, the productivity as well as the traits. So this is the most important heterosis and SCA, that is specific combining ability. We have to think of these things. These are the things I think we have to follow in the varieties as well as parental lines. In case if you are having the BT trait, that also you have to look into because that becomes the most important. And nowadays the genetic master lines are being used in the production of the hybrids, and also CGMS is also there, but still we don't have any hybrids with the cytoplasmic genetic master system. In genetic master system, female parent, male sterile, fertile, heterozygous, and male parent of the hybrids have to be involved in the maintenance breeding program as done in the conventional hybrids. Likewise, the A and B line pair and also the male parent in case of the CGMS system. This has to be, I think, done for the maintenance of the varieties and hybrids. But as far as this maintenance breeding is concerned, nowadays, it has become difficult to follow because the scheme was there, it was compulsory, people used to do it. But now I think we are not doing it. Especially, every year we are producing, developing number of hybrids and varieties and releasing and they are being notified. Hardly 5% of these varieties and hybrids are undergoing the, into the seed chain. So the question of maintaining these varieties and hybrids is almost not being done. But this is very, very important, if at all. Nowadays, the private sector is also coming forward to have the glory of their earlier hybrids with the maintenance breeding. That's what just I wanted to present here. Thank you. Thank you very much for giving me an opportunity.
Thank you, Dr. Khadib. So with this, in fact, we have finished the lead presentation. Now we have a 4 oral presentation on the selected paper. So Yeah. So already 8 minutes has given. So after 5 minutes, 5 minutes. So Dr. Bijay. D. Bijay, yeah.
Excuse me, there is a small announcement. All the poster presenters of Theme 1, serial number 26 to 49 are requested to be present at their projects as the evaluation is going on by the evaluators. Thank you.
Respected Chair, Co-Chair, distinguished delegates, good afternoon everyone. I'm going to present our work on the identification of thermotolerance diversity and unraveling its physiochemical basis in rice seedlings done at IRA. Coming to the background, global warming is a reality now, and we can see in the recent COP28, it has been already found that 1.1 degree centigrade increase in the last decade from the pre-industrial average, and it is further going to increase to 1.5 degrees centigrade by 2040 as per IPCC report. And rice is one of the more important crop that is going to be affected by this, and as per some simulation studies, even a 1 degree centigrade increase in temperature will affect the yield by 3.2 to 6.9%. And major rice growing area is Asia region, and in the Asia, if we see the temperature anomalies in the last 20 to 25 years, we can see there is a continuous increase in the higher level of the average temperature. And coming to that of the India, particularly in the months of May and June, which are the main months for the nursery sowing and the transplanting, we can see the heat wave frequency of heat waves have increased. Heat wave is nothing but the sudden rise in the temperature for a short period of 2 to 3 days. And we can see the coastal regions of Andhra Pradesh, Orissa, as well as some parts of the Bihar, UP, Punjab, and these regions, which are all the main rice belts, are going to be vulnerable for these heat waves. Coming to the rice plant, the vegetative, reproductive, and maturity, all the stages are affected by this heat wave, and mainly the germination and early seedling stages are highly vulnerable to this heat stress. Thus, there is a need to identify the thermotolerant varieties and also to induce this thermotolerance to the rice varieties, particularly in the seedling stage. And for this purpose, we have to study the entire gamut of the thermotolerance, including the basals, short-term acquired thermotolerance, and long-term acquired thermotolerance. And coming to the objectives, we have identified 2 objectives: one, to develop and validate the suitable thermotolerance diversity in seedling stage, and to understand their physiochemical basis. Coming to the materials, we have taken IR64 as a heat sensitive variety to develop or to identify this basal thermotolerance, short-term and long-term acquired thermotolerance. And we have validated this in 4 already known heat tolerant and susceptible varieties. And for physiochemical studies, we have taken the sensitive IR64 and tolerant and 22. Here we have taken, we made all these studies on the 7-day-old seedlings. Coming to the methodology, for basal thermotolerance methodology, we have to identify the temperature. Basal thermotolerance is the maximum temperature that the seedlings can tolerate, after which there will be a complete mortality will be there. So here we have identified the temperature and timeline based upon the survival percentage. And then coming to the short-term acquired thermotolerance, in case of short-term acquired thermotolerance, before giving the heat stress, a mild heat shock will be given, and after the heat shock, 2 hours gap will be given, then the heat stress will be given. That is called short-term acquired thermotolerance. If the gap is 2 days between the heat shock and the heat stress, then it is called as long-term acquired thermotolerance. And in the physiological and biochemical responses, we have seen, we have made the observation survival percentage, changes in the seed length, membrane stability index, hydrogen peroxide, MDA content, and peroxidase activity in the BT, SAT, LAT, and the acclimation temperatures of BT and SAT. Coming to the major findings, in case of basal thermotolerance, we have identified this basal thermotolerance by making a series of experiments with a temperature ranging from 35 to 50 degrees centigrade, and we have found that with increase in the temperature, the survivability has decreased, and it has reduced at 48 degrees centigrade, and further increase in temperature, there is no change in the survivability. Thus, 48 degrees centigrade has been identified as the basal thermotolerance. For this, arbitrarily, we have taken 30 minutes. Later, after identifying the temperature, we have done a series of experiments with the different time periods with this identified temperature ranging from 10 to 50 minutes, and we can see that after 30 minutes, there was no significant change in the survivability. Thus, 48 degrees centigrade and 30 minutes has been identified as the basal thermotolerance temperature for rice seedlings, and we have validated this in 4 known genotypes, particularly known tolerant genotypes and known sensitive genotypes. Here we can clearly see there is a significant difference between these tolerant and susceptible genotypes. Thus, we can use this method for screening a large germplasm for identifying the tolerant genotypes. Coming to that of the short-term acquired thermotolerance, in case of short-term acquired thermotolerance, as I told, that mild heat shock, that acclimation temperature will be given. So we have to identify that acclimation temperature and its duration. Then we have to apply the heat stress, and then we have to see the survivability. So here it has been found, after giving a series of experiments from 35 to 45 degrees centigrade, it has been observed that at 42 degrees centigrade, mild acclimation temperature has resulted in stress tolerance, both in terms of survival percentage as well as seedling length increase. When we want to finalize the time duration from 50 to 90 minutes, there was no significant effect on the survivability, but the seedling length has been significantly influenced at a 60 minutes duration, maximum seedling length has been observed. Thus, the 42 degree centigrade and 60 minutes has been identified the acclimation temperature for the short-term acquired thermotolerance. Similarly, in case of long-term acquired thermotolerance, where the gap will be for 2 days, here we have identified the 42 degree centigrade temperature and 75 minutes of duration for the long-term acquired thermotolerance. Then we have validated these in known susceptible and tolerant genotypes. In case of susceptible genotypes, we can see that if the heat stress has been given, the normal seedling percentage has been reduced from 100 to almost 10 to 20%. And that is the basal thermotolerance temperature, high heat stress. But if a mild heat shock has been given before subjecting the seedlings to this high heat stress, we can see a tremendous increase in the normal seedling percentage, both under 2 days and 2 hours duration, that is sat and lat conditions. Thus, this has been proved in both tolerant and sensitive varieties that this is very effectively, it is working, that they can be, that thermotolerance can be incorporated. Coming to the physiological response, we have seen the survival percentage. In case of survival percentage, if you see, the mild heat shock does not have any effect on the survivability, but it helped in reducing the effect of heat stress, particularly in case of SAT condition. Here we can see the tolerant variety is having a better survivability compared to that of the susceptible one. Similar thing has been found in case of seedling length, where the tolerant variety is showing a better seedling length, and membrane stability index, where the mild acclimation temperature has enhanced the membrane stability, particularly under the SAT conditions. Coming to the biochemical responses, the MDA content, the malonaldehyde content, which is considered as a very important chemical to identify the peroxidation of the lipids, including the membrane lipids, is very high under the basal thermotolerance, that is the heat stress condition. But when we have subjected them to mild acclimation temperature, this malonaldehyde content has drastically reduced under the SAT condition. So the major reason for this increased peroxidation of the lipids can be the reactive oxygen species, and we have estimated the reactive oxygen species in terms of H2O2 content, and we can see the highest H2O2 content in case of heat stress conditions, and it has been significantly reduced due to mild acclimation temperature given, which has resulted in the decreased reactive oxygen species in case of SAT condition. And the reason for this decreased ROS content can be attributed to the high levels of antioxidants, particularly we have studied the peroxidase activity. Here we can see the highest antioxidant activity is there under the SAT condition. Even the mild acclimation temperature has increased the peroxidase activity, significant increase, but when the heat stress has been imposed further, it has further enhanced the peroxidase activity, that this high quantity of these antioxidants are able to quench this ROS and thereby increase the membrane stability as well as increase the seedling performance. Coming to the lat conditions, here also we found the same kind of things, where the seedlings were able to memorize these high content of these antioxidants up to 2 days, whereas in SAT it is only for 2 hours, but here they were able to memorize up to 2 days, and a similar effect has been found. Thus, coming to the conclusions, we have identified and validated the basal thermotolerance temperature for rice seedlings, and we have identified these mild acclimation temperatures were identified, which were validated also, and these mild acclimation temperatures were effective in inducing the thermotolerance in the seedling stage. So the better survivability in case of heat tolerant variety as well as under the mild acclimation temperatures and SAT and LAT can be attributed to the high quantity of the antioxidants that have been produced, which were effectively able to quench the ROS and increase the stability and increase the seedling performance and increase the survivability. Coming to the future workup plan, we are going to continue this work by studying the genetic basis, by taking up of screening the large work, and we are going to develop a technology for by using this SAT and LAT. And with this, I want to thank you all for your kind attention, and I want to acknowledge the organizers for giving me this opportunity to present our work before you all, and also, sir, and also thank you all. Thank you one and all.
Namaste to all. Today I am going to present a small research work. This was designed based on the demand from the farmers, particularly those farmers who are interested in production of coriander for leaf purpose during summer, because during summer there is a huge demand for coriander leaves in places where the temperature rise even up to 41 degrees centigrade. So we all know that there is an effect of increase in temperature during germination. Particularly this coriander is a seed spice crop being grown during Rabi season where the temperature is. Yes sir, I am going for that. Coriander temperature germination takes germinate well at 15 to 30 degrees centigrade, but during summer the temperature goes up to 40 to 41 degrees centigrade. Based on, sir, everything is known means why I should present, sir. That is a basis for my research. That is, sir, I will explain within 7 minutes. Okay, I don't even take 8 minutes also. I will explain within 7 minutes. So the purpose of the experiment was to develop a liquid formulations using biosignaling agents as well as bioagents, so that we can provide those pre-sowing treatment formulations directly to the farmers, so those who are interested to treat and grow the crop during high temperature. So we all know that high temperatures during so to develop any liquid formulations, first we need to understand where we have to address the problem. So we all know that during high temperature there is a reduction in germination process because of reduction in enzyme activity, protein degradation, so many things will be there. To address this problem, the seed itself has a mechanism, but this mechanism is done by sacrificing the seedling growth. So we want to have a solution so that we can address the problem directly, so that we can improve the enzyme activity without sacrificing any other seedling growth or germination percentage. So in the we have tried so many signaling molecules starting from salicylic acid, nitric oxide, everything. So at last we came up only with the two molecules, that is chitosan and beta barbitric amino beta amino barbitric acid, which is a good signaling molecules. So which is enhancing the germination even at a higher temperature of 40 degrees centigrade. So we have taken this for our experiment. So we have used high vigor seed only, we have not used any low vigor seed or medium vigor seed. The purpose was not to improve the vigor at normal temperature, the purpose was to improve the vigor for high temperatures. So we have taken germination good quality high germinating seeds, and this was treated with the listed pre-sowing treatments. So for minimum of 8 hours. So we have used Trichoderma pseudomonas and also combination of both the bioagents also. And also we have tested the seedlings performance at different temperatures, like 25 degrees centigrade is a normal temperature, 35 degrees centigrade is a little higher temperature, 38 and 40 degrees centigrade. And we have conducted the experiment in, we have taken observation, we have recorded observations mainly on 3 parameters, that is germination percentage, speed of germination, seedling length and vigor index. So this is, we have taken the recorded observations on 20th. We have used cocoa peat as the medium because by 20th day we could able to see the depth of the seedlings in the between paper method. So we have used cocoa peat so that we can get the good observation, which is a basic practice followed by the farmers also. So these are the so coming to the results. So here you can say directly I can say that BA chemical at 2 millimolar concentration. So it has performed very well throughout the different temperatures. So the mean temperature, the mean germination in all temperature is 85% it has maintained. Whereas we can see that in control it has mean temperature is, mean germination is 47 and at 40 degrees centigrade. So it has already reached 100% depth of the seedlings or 100% no germination. Similarly, with the pre-sowing for speed of germination also, here though the BA chemical has reported higher germination, the speed of germination was more in biological agent treated seeds and also followed by the hydration treatment. So this is for seedling growth. Again, here also we have reported the same thing, BA chemical and also the bioagents, combination of both trichoderma and pseudomonas. So coming to the discussions, so it has been clearly seen that though chitosan has reported to have a good germination or good performance in other crops, particularly where there is a biotic stress rather than abiotic, so it has performed well in biotic stress, but it has not performed well in higher temperature stress conditions, while BA chemical has performed very well in case of higher temperature stress conditions. And also with this experiment, we have planned to design a combination of liquid formulations which is having both signaling molecule as well as the bioagents as a formulations. Thank you, sir. Yes.
Thanks, Palvi.
Sir, I have put the introduction slide so that they can read, but I am not going to read all those introduction.
Thank you. Okay, thank you, Palvi.
Thank you, sir. Thank you.
So next speaker is Swanlata Das. She is here. Dr. Swanlata Das. No. Swanlata is not present. Then the last one is Pumani. Pumani, yes. Yeah, please.
Good morning, everyone. I am Pumani, research scholar from ICR IRI, New Delhi, and I am going to present my presentation on the topic of seed priming with humic acid to mitigate heat stress in lentil seedlings by boosting antioxidant activities. So, as we know, climate change is an inevitable issue in recently, and as a result of climate change, temperature is rising beyond the threshold level, which causes irreversible damage to plant growth and development. So IPCC report says in next 2 decades, the temperature will rise exceed 1.5 degrees Celsius in next 2 decades. So this lentil is a second most important protein packed legume, which is mostly cultivated in Rabi season, and India is first rank in area and second in production. As we see, in India, Uttar Pradesh, Bihar, West Bengal and Madhya Pradesh are the 4 major lentil growing states. Here we can see the temperature is fluctuating, and it is increasing than the threshold temperature. So Indian Meteorological Department also says that 2022 is India's 5th warmest year since 1901. So it should be considered seriously so that we can sustain the lentil production in India. So okay. Yes. When the lentil seed is lentil plant is subjected to heat stress, it obviously reduces the photosynthetic activity. Sucrose metabolism is inhibited. As a result, pollen production is reduced, reproductive ability is reduced, and seed filling, seed setting, seed yield is reduced. As we know, these points are noted during the vegetative stage and the reproductive stage and maturity stage, but no seedling stage were reported. So we should, as seedling stage is very important and vulnerable to any kind of stress, so we have to focus on seedling stage so that we can mitigate the effect in seedling stage itself. So we framed a work with the objective to study the impact of heat stress on lentil seed germination and seedling development, and to understand the efficacy of heat stress, humic acid priming to mitigate heat stress impact. So why we choose to seed priming? Seed priming is one of the seed enhancement technologies which gives a faster and uniform germination, and even under the suboptimal condition, it gives a better germination than unprimed seeds. And then why we choose the humic acid? Humic acid is an organic substance and it is an eco-friendly compound, and reports also says that humic acid helps to mitigate heat stress in certain crops as well. So here is the methodology, how we did humic acid priming. We choose
Any problem?
yes, sir. Awesome. Yes, we choose the lentil seed, 3 varieties we choose, and we measure the seed weight also, and we prepared humic acid solution with the various concentration, and we found that 600 ppm humic acid for 18 hours showed higher germination and seedling parameters. So we opted this concentration for seed priming. After soaking the seeds, and we dried the seeds to original moisture content, and we kept this primed seed for germination. And 10th day we observed the parameters as well. And obviously heat stress reduced the germination in all. Here NC is the normal condition and HS is the heat stress condition. Here we see heat stress reduced the germination in all the 3 varieties, and but humic acid increased the germination by 22% nearly. And mean germination time is the time where the seeds take to germinate, and here heat stress increases the mean germination time, whereas humic acid reduces the mean germination time by somehow. And seedling vigor indices also show similar result as that of germination percentage. Heat stress reduces the seedling vigor, but humic acid priming improves the seedling vigor indices. And reactive oxygen species, malondialdehyde and membrane stability index of lentil seedlings. As we see here, hydrogen peroxide is one of the reactive oxygen species which is accumulated more in the heat stress lentil seedlings, which results in lipid peroxidation. So malondialdehyde also accumulated in the lentil seedlings, and which results in reduction of membrane integrity. So membrane stability index is reduced. But humic acid primed seeds show higher hydrogen peroxide, which is lower the hydrogen peroxide as well as membrane stability index, which helps in maintaining the membrane stability. How it is possible? Because of antioxidant activity. So as we see here, humic acid priming boosts the catalase peroxidase and superoxide dismutase content by mostly 30 to 75%. It increases. So it quenches the reactive oxygen species accumulated in the heat stress seedlings. So it helps in proper seedling development. So conclusion from this study, heat stress affected seedling germination and seedling vigor, and 600 ppm humic acid for 18 hours soaking gives better germination and seedling growth, as well as lentil seedlings produced from the humic acid primed seeds shows great membrane integrity, and by boosting the antioxidant activity, reactive oxygen species is reduced, thereby lipid peroxidation, that is malondialdehyde, is reduced. So it alleviates the harmful effect of heat stress as well. So finally, we obtained a new way to mitigate the heat stress in lentil at a seedling stage itself. So what can be further? Transcriptomic study can be done to understand the molecular mechanism of heat stress tolerance, and this study should be conducted in field level so that it can be applied properly. And as we know, primed seed has low viability. It should be study should be conducted to store this primed seed for longer time. And I must acknowledge.
Thanks.
Thank you
very much.
Thank you.
Now it's open for questions, comments, observations, suggestions, if anything. Please bring these mics to the guys.
Now that, sir, this is, I think, the first two, Madam Dr. Dadlani and Dr. Umarani. So now that the priming has been proved to be so effective, why not we make as a rule, as a condition that the Kharif 23, Kharif 24 breeder seed supplies, all of them will be primed and supplied by the recommended one. Can we make it as a policy? As a plan that that Kharif 2024, all the breeded seed that will be supplied by the NARS ICR system will be primed and supplied, so that we will have a good impact.
I got your question.
Thank you, madam.
I think what you want to suggest is that it should be mandatory that from next year onwards, all seeds should be primed.
All breeded seed to start with. All.
Yes. Yes. Yes, sir.
Should we make a recommendation for taking by government a policy decision?
Yes.
So that all the breeder seed is primed and stored.
Yes.
What is your comment?
Yes, sir. Number one, that priming technologies for all crops have not yet been standardized. Number two, the main problem of primed seed is about storability. So if you can ensure that all the breeder seeds have been primed and sown within 1 month's time,
then only it can be applicable.
I think that we can make it as a condition and then
Your
supply.
point is well taken. But the basic point is,
Yes, sir.
this is indeed a potential technology for mitigating, managing the biotic and abiotic cases as going
Yes.
on. Therefore, our suggestion would be that research should be intensified so that such technologies are perfected for all the crops.
Yes, sir.
And then see how the longevity, durability is there, and commensurate with that in the offing, there could be a policy decision.
Yes, sir.
Am I right?
Sure, I agree.
Second,
See, if you see in Kenya, they have done what is called as these balls which you prepare, seed balls. There is absolutely no problem of any viability issue.
Yes, sir.
These balls are all prepared and they kept it in gunny bags. And then they are all thrown. The entire reforestation of Kenya has been done
Yes.
by using this technology,
Yes,
balls.
yes.
But they are all basically put into the carbon balls. Because then the viability probably increases. That's how I have seen. So wonderful work which Kenyan afforestation they have already done. We can do it in a different format here.
Absolutely, absolutely, absolutely.
Good suggestion, very good suggestion.
Absolutely, sir.
Madam, there is a second question.
Then this is priming. Suppose we do like kernel, groundnut kernel, and where the radicle is protruding like an onion or in gram, will it not affect the radical in any form, especially when it is going for spinning?
You come here.
Sir, basically for priming, we should complete only the phase 1 and phase 2 of imbibition. Radical protrusion should not happen, sir. That is the duration of priming.
Already the seed is having the radicle which is protruded, after all in onion and groundnut or in gram, the radicle is just protruding out.
No, no.
It is not a
It's
round
not
seed.
radical
So
protrusion, sir.
No, sorry, what I mean is in the seed shape itself.
No, Dr. Shankaran, that is a basic principle that radicals should not protrude out.
No, sorry.
I mean, the timing has to be standardized in such a manner.
No, I'm sorry, madam. I think I am mistaken. In seed structure, in onion, gram, and groundnut, the radicle is slightly protruding out.
Yeah, it
So
is.
therefore, will it not damage in its own? Even though that we are not protruding
Sir,
the
you
radical by.
cannot say that is radical, sir. That is the embryo structure itself is like that. Elongation and division has not happened. Only if that happens, then you can find fault with priming, sir. Sir, actually, sir, for spinning, we have standardized for different, almost all the crops, commercially important crops. It varies from 1 minute to 4 minutes, sir. If you exceed that, then there will be a definite damage, sir. But the mild stress which is induced by the spinning has got some improvement over the conventional priming, sir. And we have done for vegetables also, even for onion, it is working good, sir.
Also,
Yes.
the speed of spinning is to be controlled.
The last
I think point is well taken, and I am a victim of such a situation when even priming was not being done. Harihar Ram, who is a very renowned breeder, produced lot of seed of soybean, and Kurian Sahab at that time was there, my tribute for his great contribution. We supplied that seed. Seed was loaded in gunny bags in trucks, and when it was taken and sown, not one germinated. And there was a huge cry in the country. And I was knowing what a great man Harihar Ram was. He said, sir, I tested and gave. So it is only if you are keeping 3 bags, one after another, viability will get lost. So point is well taken. This is a very sensitive issue. Please look into this. Okay. A third question, lunch पर. अरे औरों को भी तो
अरे भाई, it's a very genuine doubt, sir.
We can discuss later, Dr. Shankar.
Sir, Dr. Khadi sir, you have very nicely presented the protocol for maintenance breeding. So, just I wanted to know what is the, because we cannot go for maintenance breeding for all the varieties at a time. So, can we find out some molecular based solution that these varieties are now below threshold of this genetic purity, and then is there some work going on for that?
I think no, work is not there. Yeah, but it is required.
It is possible.
Yes, it is possible. I think it has to be done.
Yeah, because the complex protocol was very
Yeah, yeah,
difficult,
yeah.
sir. It is very
You are right,
manpower
you are right.
consuming. and
You are right.
it is- Thank you.
Very good question. Yes. And in fact, we should standardize for all the crops where you can take into the moment this climate is going to change and much more changes and swings would be the order of the day, then even somatic mutation would be far more than ever before. And therefore the variability. So where is the threshold? So we must intensify our research work, conventional as well as the biotechnical tools and techniques which are available. Okay. Yes, please. किसी को चाहिए भाई माइक? Yes.
Okay, my name is Steven Groot from the Netherlands. I'd like to ask Dr. Umarani about the spin priming. It's a very nice system, and I was surprised that you get good results. And you said it might have to do with the drying process. Have you standardized the drying, the rate of drying, the duration of drying, the humidity, or not?
So the hydro-primed seed, we are subjecting to spinning as well. And the spinning is according to the crop, size of the seed and all those things. So here we don't see what is the humidity there. We are just spinning for 1 to 4 minutes depending on the crop, and immediately we are taking it to the drying back under shade to original moisture content.
But the drying itself depends on the temperature, humidity, the speed of drying.
Definitely, sir. Yes.
Yes. That is very important.
So priming is hugely technical, sir. We accept it. So the alternative that we suggest is we have developed a seed coating formulation which gives a better improvement over germination, storage, as well as the growth of the plant and up to the yield. It is a simpler process with better advantage.
Any other question, observation, comment?
Hello.
Before thanking that, I would like to mention that the areas which are mentioned. Please recall, even the root seed coating could have been there. The 50 crore sanction which was given for Maruna Yojana, State Finance Minister from Rajasthan, would have not been achieved. Seed pelleting, aerial seeding, coinciding with the rainfall, seed would have happened. So not for the coating, pelleting, seeding, nanotechnologies, they are very, very important and a lot more investment is required. The second point which I would like to mention, that genetically tailor-making these varieties and seeds, they are not mutually exclusive. In fact, they are in complementarity, they are in association, in conjunction, so they should move forward. Third point which I would like to mention, that maintenance breeding is of paramount. As we far more pronounced changes which are likely to be the order of the day as we go along, I think much more attention is required to be paid. With these three comments, we have compensated a lot of time, and therefore we will be able to make the quick lunch decision in time. Maybe 15 minutes if you will, because we have eaten a lot of time, almost 1 hour in the afternoon. Thank you very much. Oh, thanks,
That is not vote of thanks, but we have to present the mementos for the.
Yes, sir. Sir.
All delegates are requested.